Dear students, in this module, I'm going to talk about the databases that are available for different classes of proteins. You know that CATH is a classification system based on class architecture, topology, and homology, and that it is very useful in studying the various structures as well as the overall evolution of these structures. More so, once you have such a system in place, then you can mine the data that is there for you to come to conclusions regarding the evolution of these structures. So here I will show you SCOP, or Structural Classification of Proteins, that is there as a database for you to refer in case you want to study the evolution of protein structures, their classes, their topology, as well as homology. This was built by University of Cambridge, and now you have an updated version of SCOP. So this is called the SCOP 2. You can simply look it up with the Laboratory of Molecular Biology or MRC, University of Cambridge. It is a freely available resource and it is quite useful. If you look at the SCOP database, then you will find the following statistics that are reported till date. In the database, in terms of the architecture, you have about 284 all alpha proteins. So you have about 284 proteins that only contain alpha domains, about 174 protein structures that only contain beta domains, and then you have alpha and beta, that is alpha over beta, that are about 147, and then you have alpha plus beta. So these are large, about 376, and then the rest are multi-domain or membrane and surface proteins. So essentially, most proteins are alpha plus beta, all beta, or alpha over beta. So this is the architecture that exists in the protein structures that are known. And if you want to look at the superfamily or the topology, then about 500 superfamilies exist, which are just consisting of all alpha proteins. And then in case of the all beta proteins, you have about 354, and so on and so forth. So clearly, for these four domains, you actually have lots of proteins, while these membrane proteins are few in number. Similarly, for the number of families or the proteins that are homologous, you can see that about 1055 structures are homologous, or that they belong to the different families, and you have alpha plus beta for this. Okay, so having looked at these statistics, there are different databases that are available other than the CATH or the SCOP, and these include the FSSP, or the family of structurally similar proteins. So the algorithm they use is the Daly algorithm, and we'll study that later. Also, there is the P-class, or the protein classification, and it is based on the LOC and 3D search algorithm.